Welcome to our NF Knowledge Series webinar for May 22. It's a very special day. This is NF2-Related Schwannomatosis Awareness Day in the midst of NF Awareness Day, and we're thrilled to have you here. And I'm very excited to present some very special information to this community on this day. So if you don't know me yet, my name is Kate. I work with CTF and I am your host for these webinars. So I'm very pleased today to be welcoming two wonderful people that I get to work with every single day. Dr. Annette Bakker is our CEO here at CTF and a brilliant scientist who has done incredible work in this field of driving research forward and pushing us toward real treatments for the patients and families in the NF community. So we're thrilled to have her joining us today. She will also be joined by Dr. Marco Nievo. Marco is the CSO for CTF Europe. He's based in Italy and has definitely a European at heart. Marco has been with CTF for over a decade and plays a very important role with the inception and creation of CTF Europe. So they're both really fantastic scientists and they have some great information for you guys today. So I am going to hand things off now to Annette.
Thank you. Thank you so much, Kate. First of all, welcome everybody to the NF2-Related Schwannomatosis Awareness Day. There's a couple of things I wanted to mention before I hand it over to Marco to dive into the science. I want to make sure you know how committed we are to NF2. And I know you will all think you have 2 approvals for NF1, but we have a lot of great data now, and we're pushing to also have approvals for schwannomatosis. And I just want to indicate how difficult it is to get a first drug approved for a new indication. And AstraZeneca has really paved the road, or Alexion, for the first drug approved for NF1, and especially for plexiform neurofibroma in NF1. But I can assure you that we're working really hard to make sure that we get that same success for NF2. The second thing, for the mathematicians in the room, you will see that Marco is mentioning that we're funding 15 projects and he will report on 13. So there are a few projects that are incredibly important and that are percolating. We sometimes cannot talk about them for confidentiality reasons, but we will be talking about them in the very near future and as soon as we are allowed. Confidentiality means hope. So I just want to make sure that you understand my indication here. We are pushing for drugs, we're pushing for targets, and as you all know, you first have to understand the mechanism, then you identify the drugs, then you de-risk the drugs, and then you bring them to the clinic. So this is a whole pipeline, and that is what Marco will show you, that we have a very busy pipeline. We have drugs that are ready actually to go into the clinic. But I'm not going to steal Marco's thunder. Marco, I'm going to hand it over to you as a great medicinal chemist, patent attorney, CSO of CTF Europe. I can keep going. You have a lot of titles, but today what you are is our presenter. Go, take it forward, Marco.
Thank you. Thank you, Annette. Just 1 second. I need to share my screen. And I need an okay from Kate.
Yep, we can see it. It looks great.
Great. So welcome, everybody. I'm really honored to actually give you, to go through what we are actively funding at the moment. So the NF2 specific portfolio consists of 14 active grants, not 15, sorry, Annette. And this represents an investment of $2.5 million. Some of you may know already that, some of you may not. So the categories of grants that CTF awards fall into 4 categories, basically. The first one is called the Young Investigators Awards. And these grants are designed to allow very young researchers to actually establish themselves as established researchers in the field so that they can continue to thrive and research on NF, or in NF2 in this particular case, and build their career around this. There is another category of grant that is very focused on drug development, which is called the Drug Discovery Initiative, which is about testing drugs, testing potential drugs in animal models or in cell models to either progress them to the clinic further or kill them, because you actually need to understand if something works or doesn't work in these what we call preclinical settings before actually venturing into man. There are also clinical research awards, which are designed to help small pilot studies that can actually be then transformed into a full bang clinical trial. And then we have contract awards, which are awards that don't fall into the other three categories, and which some researchers approach us with specific projects that don't fall strictly into these categories, that may require a lot of funding in many instances, and so these go through a review on our side, of course, and then they get awarded. We also have, I mean, for contract awards, we also proactively, the science team at CTF actually proactively reaches out to researchers when we understand that some research results need a follow-up to bring them closer to the clinic, to bring them to the patients. So we actually contact the researchers ourselves and understand with them and design studies that will actually progress compounds or concepts closer to the patients. So I will start by actually describing two clinical awards, the two NF2 clinical awards that are currently being funded. Yes. These are actually related to quality of life studies, because it's very important to research, you know, basic science, test drugs in animals, etc. But then there's the day-to-day life of NF2 patients. And in the day-to-day life of NF2 patients, you need solid science to actually measure their quality of life so that you can understand how to improve their quality of life. So the first study is actually carried out by Vanessa Bakker at NGH. And it is about, because the quality of life questionnaires that are currently used in clinical trials or in general are not very specific to NF2. So this project is about designing a quality of life questionnaires that is specific to NF2 patients. It goes through iterative processes of identifying all possible measurements that actually can be used to measure the quality of life, and submit this to the patients as well as to the doctors, and narrow down these to those that are truly specific. Currently, Vanessa Bakker actually allowed us to share some results, and it's actually coming to the final stages at the moment. And so the questionnaire is almost ready, and it's undergoing now the final iterative process of validating with the patient, validation with the patients. And actually, there is a call from Vanessa Bakker if there are any patients interested in participating in the study, to actually enroll. And to do that, maybe the best way would be to actually contact Kate at the end of the webinar, and she can put you in contact with Vanessa to see if you can or will enroll. Another study, which is led by Konstantina Stankovic at Stanford University, is about biomarkers. So for those who don't know what a biomarker is, it's actually a measure that tells you something. A biomarker is a measure that is indicative of something else. So she has identified, before applying for the grant, she had identified biomarkers which are proteins that you can measure in the blood, that are indicative of the presence of a tumor, and are indicative of the presence of hearing loss. So her question now is, do variations, do changes over time in these biomarkers, could these be correlated to further progression of the hearing loss or further growth of the tumor? So if this study actually ends up identifying biomarkers that can be related to this should improve the quality of life of patients, because the indications that their tumors are growing or that their hearing is actually decreasing could be done via a simple blood test instead of undergoing an MRI and instead of undergoing a whole audiometry testing. Now, going on to basic biology studies. Now, you know that the research, the process to actually bring a drug to the clinic starts from the basic biology, where you try to identify mechanisms that are actually that are responsible for the tumor growth or for the hearing loss, and that actually so that you identify what we call molecular targets. You identify things that could be druggable and could be actionable upon. Typically, these are all Young Investigators Awards. So the first study is by Sarah Vega at MGH again. And she is studying the microenvironment. Now, the microenvironment is a very hot topic in tumors in general, in cancer and in NF2 and in NF2 as well, because the tumor exists in a context, and in this context, you have different kind of cells that actually interact with the tumor, and it's very important to study how these cells interact and why these cells interact with the tumors, because in many instances, like in this one, there is a particular class of cells called macrophages that actually protect the tumor from being attacked by the patient's own immune system. So her research is aimed at identifying really the molecular mechanisms that help that turn these macrophages into protectors of the tumor rather than protectors of the body in general. So the potential impact here is to identify druggable mechanisms that target the microenvironment, so that you can target the tumor itself with a drug that attacks the tumor, but you can also attack the microenvironment that helps this tumor thrive. Yes, ISAM at the University of California is actually analyzing all the proteins found in the inner fluids of two specific NF2 mouse models. And by doing this, it should be able to identify potential biomarkers of hearing loss and also potential druggable mechanism. This study is very, very cleverly designed because he's using two mouse models to do that. One mouse model, which has an NF2 mutation and which actually develops tumors as well as and develops acoustic vestibular schwannomas and experience hearing loss, and another model where the mice do not develop tumors in the auditory nerve, but they do experience hearing loss as well. This is a very, very important study because it is not yet very clear how much the hearing loss is connected to the growth of the tumors. So thanks to this study, we will have more insights into how to separate the two mechanisms. Are we having hearing loss because there is a tumor, or is the hearing loss somehow completely unrelated or only partially related to the presence of the tumor? The next one is about, angiogenesis. So Shirupa at MGH as well is studying a protein called apelin. Now, angiogenesis, for those who are not scientists, is the mechanism, the biological mechanism that actually builds blood vessels. And, you know, tumors are fed by blood vessels. And a drug that is currently used by many NF2 patients is called bevacizumab, or Avastin. It's the same compound, it's just a different name for the same thing. Is used in many patients to stop this mechanism of angiogenesis, because if you stop the blood vessels from developing within the tumor starves and should die. The issue here is that Avastin doesn't work for all patients, and Avastin for some patients stops working at some time. Now, you can address angiogenesis by through many different mechanisms. So Avastin actually acts on a compound called VEGF, but this is not the only way that tumors build vessels. So her work focuses on this alternative mechanism for angiogenesis called apelin. And so her work by analyzing the biology around and how apelin actually affects the tumor growth in animal models will help understand if there's an alternative mechanism to Avastin to actually block the angiogenesis in tumors. The next one is from Anna Nagel at the University of Central Florida. And this, again, is a very, very constructive way of building on previous results. There was a drug called CUDC-907, which is known in both in cell and animal models to induce cell death in NF2 cells. But the problem with that drug is that it is way too toxic to be administered to NF2 patients. It's a drug that has a toxicity profile that is not compatible for an NF2 patient. It would affect their quality of life way too much. It's a drug that's being developed for cancer, and in a cancer setting, one tends to tolerate side effects a lot more. I mean, not to tolerate them, but to accept side effects a lot more. So instead of saying, okay, so she said, yeah, CUDC cannot really be used as a drug in the future. But because we know that CUDC works, we are going to use CUDC to understand what exactly does CUDC in the cells to provoke the cell death. And through that, she may be able to identify new mechanisms that actually contribute to the cell death. And these new mechanisms potentially could be targeted by drugs that have a much better toxicity profile. Now, jumping on to the next phase of the drug discovery process. So now we've been talking about lots of basic biology, understanding mechanisms, identifying potential mechanisms that can be targeted by drugs. The next step is then to start to test drugs in these models, to test proper potential drugs in these, to see if they can go into men later on. So in this case, again, Avastin. As I said before, Avastin has this, it doesn't work for all patients. Some patients unfortunately progress after a while they're on Avastin. And because she found this, other researchers had identified a drug called beltuzifan that can work in patients where Avastin has failed. So she wanted to investigate. She said, well, it's exactly the same situation in NF2. Avastin fails, and let's see if beltuzifan can actually be used in NF2 as well. So she is now doing studies in mice to understand if beltuzifan could be an alternative to Avastin or a complementary therapy to Avastin. And we know that, you know, Avastin also affects the hearing. I mean, Avastin has a lot of a positive impact on the hearing of patients. So she is studying both the effect on the tumor growth and on the hearing. Another one is Larry Sherman from the Oregon University. Now, there is some basic biology findings that platelet aggregation, which is a very technical word for blood clotting. Platelets are components of the blood, our blood cells that actually help the clotting and the healing of wounds. And it was found that there is a lot of these platelet aggregation in vestibular schwannomas. So because there is a protein called thrombopoietin that is majorly responsible for this platelet aggregation mechanism, he is studying whether a drug called TPO-AZO could actually, because it inhibits this thrombopoietin, could actually have a beneficial effect in an NF2 setting as well. Dominique Lallemand, who works in France. So this is a very nice example of what the drug discovery process is. What you do in drug discovery, typically, you don't immediately find a drug. You find what is called a hit. And this hit has characteristics that make it promising as a drug. But these hits need to be worked on chemically. I mean, you chemically modify them so that they maintain their activity, but they acquire more drug-like properties. So one very, very hot molecular target in NF2 is called the Hippo pathway. And in this Hippo pathway is a complex called the YAP-TIAD complex that actually plays a key role in the proliferation, in initiating and maintaining the proliferation of tumor cells, so the growth of the tumors. So they had identified hits, which are peptides that disrupt this complex and thus block the Hippo pathway and thus block the proliferation of tumors. But these peptides are too quickly degraded by an organism. So his work is about modifying these peptides to make them more resistant to degradation in the organism whilst maintaining the activity. So doing this hit to lead a process, moving a compound closer to actually being a drug. And so he is studying this and doing modifications on these peptides to make sure that these compounds could actually one day transform into a drug. Now, this is a very complicated study to explain, at least. So in very lay terms, many NF2 patients have what we call truncating mutations. So imagine this is the normal NF2 gene, and a patient with a truncating mutation will have at some point actually produces just a little piece of the protein, because there is a mutation in the gene that says stop producing the protein at this level. So instead of producing a protein that has this length, it will produce a protein that has this length. This protein usually is defective, which leads to the NF2 symptom. So her hypothesis here was to say, okay, I have this truncating mutation here, but what if I just skip skip that truncating mutation, so that I get a protein that, if this is the NF2 protein, instead of getting this little piece, okay, you actually get a shorter NF2 protein that only misses this bit which is being skipped. And to see if these proteins that are not the NF2 protein itself, they're just a version of the normal NF2 protein with just a bit missing at some point in the chain, and to see if these actually fit the bill and do the job that NF2 does. And so to do that, she uses a technology that is called exon skipping. And she has shared results, and she has agreed that we share results. We share some of the results with you today, and the results are very positive. She has managed to, so through this technology, she has analyzed what we call NF2 modified protein that misses just that little bit. So this protein, they actually produce an NF2, a modified NF2 that actually works, and it works by stopping the proliferation, by reducing the proliferation of cells. Now on to the clinical trials, because once a drug has been tested in preclinical models and the results are promising, then the compound gets moved into clinical trials. So here we have three very, very exciting projects. The first one is the Init Trial, which is led by Scott Plotkin at MGH. And it's a very cleverly designed trial, very innovative trial. There are very, very few trials of this kind ongoing in the world. And it's a trial that actually is innovative because many drugs, just the architecture of the logistics around the trial make it possible to test many drugs in parallel. So the first drug tested was brigatinib, and the results were quite impressive because they shrink 10% of tumors that are growing, and 23% of all tumors in general. Now, there's a second drug that is being tested called neratinib, and that has up to now enrolled 20 patients. Now, for how the protocol is structured, this milestone of 20 patients is very important, because this is where the Init team does what we call the interim analysis. So they stop enrolling patients, and they analyze the data acquired on these 20 patients. And Scott Plotkin will be presenting data of this interim analysis at the upcoming conference in Washington, D.C. At the end of June. And we are all very much looking forward to actually seeing these interim results. Another study that we are funding is led by Oliver Hanneman in Plymouth in the United Kingdom. So this is based on the fact that we all have in our genetic setting, in our DNA, we all have what we call endogenous retroviruses. And these are retroviruses that are normally dormant in cells, in normal cells. But there is a specific retrovirus called HER-VK that actually is overexpressed in NF2 cells, and that actually drives the tumor growth. Now, there are very common HIV medications that actually inhibit HER-VK. So Oliver and his team, actually, they designed what we call a phase 0 trial. Very clever design, where they will test. So these drugs that are known in HIV have been used for ages by HIV patients, by thousands of HIV patients in the world. So we know what the toxicities are. We know exactly. In administering these drugs to patients, you won't have any surprise in terms of toxicity. So this allows you, in the UK at least, to design trials in humans that are on very few patients. And so in this case, it's only going to test 8 patients. These 8 patients are being selected because they have cutaneous schwannomas. And the reason for these cutaneous schwannomas, for selecting these patients, is that the trial, so the patients will be administered the drug, but they will be biopsied and try to understand if the drug actually has a biological effect on the tumors. So you have to, in order to minimize the impact on the patients, he has selected patients that have cutaneous schwannomas, very easy to biopsy. And so here again, enrollment has started, and we hope to have results quite soon. But this is a very cleverly designed trial, because if the results are positive from these 8 patients, this will allow and speed up actually the implementation of a large scale study. And the last one is a group in Korea that have actually identified a very unusual and very out-of-the-box mechanism that could be targeted in NF2. So again, a bit like the HER-VK, in our cells, we all express an inhibitor, a generic kinase inhibitor that is called RKIP. And this RKIP regulates in normal cells, regulates tumor growth, because it acts as what you call an oncorepressor, because it tends to, if there's an overactivation of these kinases, which are typically found in tumors and cancers, RKIP inhibits them and maintains the normal state of the cells, and helps maintaining the normal state of the cells. And they have found that this compound called PLGN01 actually acts as and so in NF2 tumors, RKIP is underexpressed. So their hypothesis was to say, what if we manage to reestablish normal levels of RKIP in a tumor cells, so that RKIP does its job, which is to maintain the cells and make sure that they don't grow like tumors. So they have found this compound which actually indirectly induces the expression of RKIP, and they have tested these in animal models, and the results were quite impressive. So they have moved on to doing what is called a phase 1/2 trial. And they are, so phase 1/2 trials are designed to actually find a dose. And so to find the right dose to administer to the patients, that doesn't have to, the idea is to give the maximal dose that has tolerable toxicities and side effects, and at the same time has some effect on the tumors. So they are doing it's called so they increase the dose. They take 5 patients who will have 1 dose. They analyze what the situation is in terms of side effects in these 5 patients, and they decide whether or not to increase to a higher level of the drug. Really, in these trials, the effect, okay, is only indicative. What you want to do there is to just identify the right dose for the patients. but they are also monitoring the patients for the effect, okay, just to see if they see some effect in this trial setting. So they will actually be presenting as well at the conference. Apparently, they are seeing effect in the patients. And this is quite astonishing because the patients are only being treated for a month in these dose escalation. You just, for 1 month, you see if they tolerate, if there are no side effects, then you move on to the next. But they seem to, and because they were monitoring, of course, the patients by MRI to see how the tumors were reacting, they will be presenting data at the next NF conference in Washington, D.C. So I am actually done with this presentation. I hope I've given you a nice, overview of the investments that CTF is making all across the board, from quality of life to basic biology, to understanding if a drug can actually make it to man, to how and to how the drugs are behaving in man.
So first of all, thank you so much, Marco, for this nice layout of the situation. So the first question that I have to the community here is, do you have any questions? Everything that we have been able to present today and everything that we have been able to do at CTF is also with a big thanks to everybody's contribution. So thank you for your financial contribution, for being a patient, for being willing to enroll in clinical trials. So I just want to make sure that we never forget anyone who is on the call here. But we haven't seen any questions coming in yet. But if you have any questions, please don't be shy. Ask the questions, and then I will jump in and talk about tomorrow if there is no more questions.
Yeah, we're not seeing anything coming in, so I guess you did a really great job, Marco. Thank you.
Or a terrible one.
So what about tomorrow? So you have heard Marco talk about brigatinib that is actually really active in the patients. We are working really hard because this drug is approved and could theoretically and is being prescribed what they call off-label. But in order to get it reimbursed by your insurance companies, we want to make sure that it's either approved or that we can get that into what they call NCCN, which are in fact treatment guidelines, so that the clinicians can prescribe it and the NF2 patients get reimbursement. So that is one aspect that we are working on really hard together with Scott Plotkin and Jayshree Blakely to figure out what we can do to make brigatinib reimbursable. The second thing that I want to say is that for Intuit, as Marco described, this is a very unique, what they call platform basket trial, which is a trial that really allows us to keep that trial open for 10 years and where companies can come in and literally plug their drug into the trial. So as you can imagine, a huge time saver over the usual clinical trial where you have to start building the trial from scratch before you can even enroll the first patient and bring in the first drug. We are, for the moment, we can't disclose because we're in full communications with the different companies, but we're actually looking at 5 different compounds or compound combinations that could enter into this clinical trial. So needless to say that we are fundraising for this to make sure that these clinical trials can happen. But it's a huge hope, I hope, for all of you to know that we're actually in figuring out what the next possible candidate could be. Brigatinib, neratinib, and what are those other? And there is like 5 that are lining up here. As Marco indicated, the Korean company is a company that we identified and that I think is a huge potential. And as you have heard from Marco, they will be presenting at the conference together with all the other incredible discoveries that have been made, from new biology to targets to quality of life and all the way to clinical trials. So we can only encourage you to be part of our amazing conference. But as you can see, a lot of the important things are happening. I could imagine that some of you may wonder why we're not mentioning any of the AAV gene therapy work that we have heavily invested in, because we believed in it, like so many other of our colleagues in the nonprofit world and in the for-profit world even. But you may have seen even on, things like Fierce Biotech or Endpoints or all these news channels, is that many companies for the moment are stopping AAV because of toxicities, because of maybe not reaching that many cells that they were hoping to reach. So it's maybe not as efficient as a gene therapy for certain indications. And as hopeful as we were that we identified this group in Australia that was able to identify AAVs, which are these delivery systems for gene therapy that would not go to the liver. So we were very excited about that, because that would manage toxicity so much better. And we invested pretty heavily in NF2 loaded of these special AAVs. But what they call transduction, or let's say the arrival of this vehicle with NF2 in the cells where it really were supposed to get, were not as great as we were hoping. So in agreement with the researchers, we actually discontinued that project. So for the moment, we're looking at something else that we can talk about, but that we hope we will be able to talk about very soon. We're not just looking at it, we're actually working on it, but we cannot disclose anything at this point. But just want to make sure that you know that we're pushing, and we hope you join us in pushing. Thank you so much. And I'm going to hand it over to Kate now.
Perfect. Excellent. Thank you both. Excellent presentation. And I have some housekeeping, but it also addresses just a couple of questions that came in while Annette was wrapping us up. So the conference that we're talking about, there are two events happening this summer in Washington, D.C. The NF Summit is our conference for patients and families affected by any form of NF. If you are not familiar with it, or if you have not registered yet, I want to encourage you to consider joining us. It's not too late. Our room block for our special discounted hotel rooms does end on May 27th, so we would love to see you there. And as some incentive, I'll say that a few highlights for this particular community is that the summit agenda includes sessions on imaging guidelines, several sessions on navigating hearing loss and communication around deafness. Dr. Rob Avery will be speaking about ophthalmic, so eye issues that happen in children, both with NF1, but also with NF2-related schwannomatosis. We'll also have several sessions on clinical trial participation and how to get engaged as a patient or a caregiver in research development. So it's going to be a really fantastic time and a great way to connect with the NF community and to learn more about what's happening. The other conference that Annette and Marco referenced is our Medical and Research Conference, which is also happening in Washington, D.C. It kind of starts up right as the NF Summit is winding down, and there are always some really great and exciting things come out of that as well. So our next webinar, I want to warn everyone, we are skipping June because of our big meetings, but we will be back on July 10th talking about AI. This particular webinar is going to talk first about facilitating the learning behind AI and machine learning, but also the benefits and weaknesses of this, as well as the specific impact that it might have for NF in particular. I'm really looking forward. I've heard this presentation before, and I don't think you'll want to miss it. For someone like me who is not a tech AI person, it was really interesting and accessible as we think about how it might impact not only research, but also our healthcare. So for a list of dates for all of our future webinars, be sure to visit our educational events calendar. I know that Cindy's just dropped a link to that, but you can find all of our resources by visiting ctf.org and just poking around. It's a pretty navigatable website. It's not too hard to find the different things you might be looking for. We have a page there that's dedicated to NF2-related schwannomatosis, and we regularly update that with things that are happening in the research and funding world. And you can also get signed up for our newsletter. In particular, we have an NF2-related schwannomatosis newsletter that goes out every month that you can specifically sign up to receive. And that's always full of great information as well. If you have questions about this webinar, or if you asked questions today that did not get addressed in the conversation, please don't hesitate to reach out to us directly. You can reach us at webinars@ctf.org, or you can also just reach out to me directly. My name is Kate, and I've put my email in the chat for everybody. So please feel free to send me an email. I'm happy to speak with you. So that's all of our housekeeping. I know that's a lot of information. I want to say again, a big thank you to both of our speakers, to our wonderful captioner who is here with us today, and to all of you in the community who joined us to listen and to learn. We hope that, as Annette said, that this information is exciting and motivating as we continue to push forward for a better future for everyone that's part of this community. So thank you for being here, and please join us again in July, and I hope to see some of you in Washington, D.C. in June. Thank you, everyone, and have a wonderful
Kate, can I mention one quick thing? Is there is a streaming component as well, right, to the conference?
Yes, there is. The NF Summit is in person only. We do record everything that happens there and make it publicly available on the CTF's YouTube page within a few days of the event happening. The NF conference does have a live stream option. And if you're curious about that as a patient or caregiver, I'd love to speak with you and just talk about sort of the goals for the conference and what you may or may not be able to get out of it. But please don't hesitate to reach out and ask me if you would like more information about that. Okay. All right. Well, thank you everybody for being here, and hopefully we'll see you again soon. Have a wonderful day. Bye, everyone.